Nirenberg and Matthaei were the first scientists to determine that a specific RNA sequence coded for a specific amino acid. For their procedure, Nirenberg and Matthaei put together poly-U, a strand of RNA only consisting of uracil nucleotides, and 20 amino acids, including one radioactive one, all in a cell-free environment. They would incubate the tube this mixture was held in, filter out the protein, and check for radioactivity, and then repeat the procedure for the remaining 19 amino acids. In one tube which had a radioactively labeled protein, they found newly synthesized protein made from the amino acids coded by the poly-U. From this, Nirenberg and Matthaei were able to identify three major codons. The first they identified was UUU, which codes for phenylalanine. Eventually, they used poly-C to find CCC, which codes for proline, and poly-A to find AAA, which codes for lysine. The codon GGG does not work to code for amino acids and therefore isn't valid unlike the other three, which are. Along with this information, Nirenberg and Matthaei were able to determine that a triplet of nucleotides is what would make up a codon and that those individual nucleotides could be changed to code for different amino acids. Nirenberg and Lieder had a similar experiment, and they were responsible for what's known as the triplet binding assay. Their procedure consisted of putting 20 charged tRNAs, including one radioactive amino acid, ribosomes, and very short mRNAs that each had one known codon through a filter. They would allow for binding to occur between the mRNAs and some of the tRNAs, and then filter out whatever hadn't binded. After which, the filter paper was checked to determine whether or not it was radioactive. What this meant is that the filter paper would become radioactive if a particular trinucleotide coded for the radioactive amino acid. So, for example, the mRNA with GUU was used, and on the filter the tRNAs were bound to was valine. This meant that the GUU codes for valine. From their experiments, Nirenberg and Lieder were able to identify 61 codons, all of which code for at least one of the essential 20 amino acids.